Okay, so Assalamu alaikum everyone. This is the final part in the epigenetic cell memory, and we are going to talk about a very interesting phenomenon, which is basically imprinted genes. This is, going from parents to offspring. So memory of gene expressions, which are imprinted by parents, and the state of genes on or off, which is marked in parental gametes, is passed on to embryo, to offsprings, and they maintain that state. I try to draw what happens during gametogenesis, or process of gamete formation. For example, so we have the primordial germ cells, and we have on the y-axis we have methylation. So high degree of methylation to almost little to none here. And on the x-axis we have the time of development. Here we have these horizontal lines. This time point is actually representing the fertilization, the event of fertilization. What happens? The primordial germ cell, which is a diploid cell, it undergoes meiosis and eventually gives us mature oocyte or spermatocyte, and then we have, after fertilization, embryonic day 5, embryonic day 10, etc. And on this axis we have the development going on. In oocytes or in spermatocytes, there are many genes which do not contain any methylation. So we will have them here, like the genes which are non-methylated are these ones, okay? Non-methylated genes. In the primordial germ cells, even after undergoing meiosis, so they remain non-methylated. But then there are groups of genes which are methylated, and what happens when primordial germline cells, they undergo meiosis, there is a wave of demethylation, which is followed by remethylation, and then there is a big wave of sharp demethylation, and then they come back again. Here the blue one is the paternal gene, the father's gene. Similarly, if we talk about the maternal genome, so similar pattern in the oocyte takes place like this. Then methyl marks are reset again, but as compared to the father, there is delayed demethylation in mother, and then remethylation goes like this. I said we are going to talk in particular the imprinted genes, which color I should choose? Let me choose the color so that Okay, let's choose the green one. So you will have a class of genes which will, during process of gamete formation, once they get remethylated, they remain methylated like this. These are, the green ones are the imprinted genes, and this was the overall paternal genome, and pink one is the maternal genome. Now, why there is a class of genes which we call imprinted genes? These are 200 or so genes in our cells which are expressed in a very characteristic manner. We call them parent of origin gene expression. Parent of origin means, for example, I give you a small example here. For example, we have two genes. One is called insulin growth factor 2. The other one is called H19. This is father's copy. Let's use the same color like the paternal genome we used. And the mother's copy is also indeed IGF2 and H19. But what happens in every human, that IGF2 is expressed only from father. So the father's copy which zygote is going to inherit, only father's IGF2 is expressed. And mother's copy is off. And father's H19 is off and mother's H19 is on. So this is what we call parent of origin gene expression. We all have only our father's copy of IGF2 will be on and mother's H19 will be on, mother's IGF2 is off. Now, let's come back here. This is a somatic cell. This is a primordial germ cells, PGC. It's a diploid cell. It contains this information. But this information needs to be erased before mother or father, they are going to pass their IGF2 copies to the zygote. Here, whether you are female gamete, female PGC or male primordial germ cell, the expression pattern is going to be like this. IGF2. So the mother is having her father's copy of IGF2 on. Father is also going to have his father's IGF2 on, but when they are going to pass on, both of these copies in this diploid cell here, they are going to be considered mother's copies when they are going to come here in the gamete, in the zygote. So what we do, the primordial germ cells, they undergo massive demethylation. All the methylation is erased and the imprints are reset. Here, for example, you see here in case of IGF2 in mother, mother is going to methylate IGF2 to keep it silent. Father is going to unmethylate both the copies because this diploid cell is going to give you 4 sperm cells. Each sperm cell should contain IGF2 in the on state. No methylation, no imprint. Okay? And mother's copy should be having IGF2 in the off state. Here, father is going to have H19 in the off state. And all the copies, all 4 sperms, they should have both H19s in the off state. Okay. Once the imprints are set, here when fertilization takes place, sperm and the oocyte fuse together. When zygote grows, here there is no change or reversal of methyl marks on these imprinted genes. There are 200 or so loci in our cells. What happens that because here you run the danger of losing the imprints. But the other genes, the other regions in the genome which are methylated, the father's copy, and this is a very active demethylation. A reversal of all the methyl marks, almost all methylation is gone except the imprinted loci. And then methyl marks are reset. This is like constitutive heterochromatin, or if there are some genes which are silent in the PGCs at the oocyte or spermatocyte formation. Here, the DNA methyl mark on other loci other than imprinted, they are reset. So that zygote is starting its life afresh without any history. It tries to, be a very healthy baby according to the programmed DNA methylation or the epigenetics, whatever the epigenetics of development are. Mother's genome is known to be, slowly undergoing demethylation and then it comes back. Okay? But once they are set, whether it's constitutive heterochromatin, and then these methyl marks, they are, when the differential gene expression kicks off, the genes which are going to be switched off, like eye-specific genes are switched off in heart cells, and vice versa. Heart-specific genes are switched off in eye or neurons. So they will be differentially methylated in neurons, in eyes. They will be switched off so that the heart fate is gone, is blocked in neurons or in eye, or in the eye cells you have heart-specific genes switched off by DNA methylation. DNA methylation, as I told you, is maintained through maintenance methyltransferase, and these differential methyl marks or these differential gene expression patterns are inherited generation after generation. This was very important to learn, that epigenetic cell memory is not only about maintaining or maintenance of cell fates which are established early during our development, but there is also maintenance of these cell memory marks, which is a DNA methylation or covalent modifications of histones, which are linked to imprinted genes, because this is going from one generation, the parents, to offspring, and it involves meiosis, inheritance of meiosis. So here you can see IGF2, the sequence at DNA sequence level, both IGF2, father's copy and mother's copy, they both are same. There's no change in the DNA sequence, but due to DNA methylation or covalent modifications of histones, you have selective silencing, parent of origin gene expression. There are other examples of role of epigenetics, and another good example which we all know it from our courses in sophomore or so is the X inactivation. X chromosome is randomly inactivated. Either of the X, whether the X in human females, human females, they have two X chromosomes as compared to males. So one of the two X chromosomes become totally switched off or inactive. We call this X inactivation. And since the females, they have one copy of X coming from one chromosome from father, the other from mother, the gene dosage becomes unequal as compared to males. So nature has evolved that one of the two X chromosomes is completely, jumbled up in, and we call this bar body. So this once one of the two X chromosome becomes off, all the genes become transcriptionally off. Through mitosis, this X chromosome is going to remain switched off in all the cells, progenitor cells of this mother cell. Now, you will be surprised that X inactivation in mammals is also due to DNA methylation, and it should not surprise you if I bring in polycomb. So enhancer of zest, PRC1 complex, PRC2 complex, enhancer of zest, chromodomain containing polycomb, the ring protein, etc. All these PRC1, PRC2 complexes which were found in flies, they are found to maintain inactive X chromosome throughout mitotic division, cell divisions during development in human females. We have here, I'm talking about, loss of DNA methylation in humans. You can think of in plants as well. Transposons are maintained in the off state because they can play havoc with our genome stability. And transposons, all the transposons, they are highly methylated, DNA methylated, or histone lysine 9 trimethylated. And they are also kept under tight control, and epigenetic phenomenon, they are involved. So epigenetics is something which plays very crucial role in our development, because more than billion cells, which are divided in 200 different cell types in our body, they contain same chromosomal information, same genes, yet they give you different identity, different fates. And these fates, they need to be maintained. If my skin cell forgets its identity that it is a skin cell, it will undergo in de-differentiation, uncontrolled proliferation, and cancer will develop. So a number of polycomb proteins are also known as tumor suppressors because they maintain the cell fate. They control, they act as tumor suppressors, they control cell cycle as well. So with that, I would also like to end by recommending you to read, papers, and these papers are from Stephen Ree. We mentioned briefly. It was Nature 2002, and also read, I think it was Jürg Müller was the first author, if I remember correctly. It came in 2003, I believe. I may be wrong here. And it was Cell paper. So Stephen Ree's paper is about AH3K9 trimethylation, the SUA 3-9. Jürg Müller's paper is about EZ, enhancer of zest, so AH3 lysine 27 trimethylation. And there was also a paper, back-to-back papers in the same issue by Vince Peroda's lab about EZ. And also there was a paper from, I think, if I remember the correct name, Kao et al. I think it was Yi Zhang's lab in Science, which linked X chromosome inactivation, I believe. So these three papers will give you very good understanding how epigenetics is involved in development here, constitutive heterochromatin here, epigenetic cell memory, here, X chromosome inactivation. And in these days, as we are talking, epigenetics is now, one of the fundamental principles of maintaining the pluripotency of stem cells, controlling, normal development of cells. I told you act as mechanism to block cancer progression or tumor suppression. So I think the more you read about these three papers, I'll share them on LMS, and I would expect that you should read these three papers so that your understanding about role of epigenetics in development, it becomes more clear. So I hope you will enjoy these lectures. I would like you to send me any questions, detailed feedback if you have any, on my email. Thank you very much.